Okay, so I'm going to not go on for quite as long today, so you guys will get an early finish. You can go and do some fun things in New York, whatever you want to do. What I'm going to talk about today is basically some sort of practical considerations when you come to start analyzing genomic data. And we're going to cover a number of different sort of topics that I think are important, things like data quality control and some methods you can use to start looking at that and figuring out if there are major problems in your data, even before you start doing any analysis on it. Talking about things like batch effects, how you can avoid them. Very important things like how to visualize your data in ways that can give you sort of insights into what's going on. Again, very important when you start to do any kind of analysis. We'll cover some ways of plotting data that hopefully some of you know about already, but I'll try and point out how they can be much better than some of the more traditional methods. We'll cover some things like power calculations just briefly, and then finish up on things like p-values and what do they mean, and point out some of the oddities that can occur with the genome and when you're talking about p-values and how to calculate them. Let's start off with sort of data QC. So a lot of people think, say you were to run an array experiment, a sequencing experiment, whatever, you get your data and, if you don't know what you're doing, probably the first thing you want to do is say, all right, how can I throw this into some pipeline and start analyzing it? That's not what you want to do. The first thing you want to do is understand what the data you have is, because often there can be some major things wrong that will fundamentally change how you want to go about approaching that data. So just for example, probably every large cohort study I've ever worked on always has some sample mixes in. Like, people study 100 or 1,000 samples, and I can almost guarantee that you'll have a sample labeled X, and it's not X, it's Y, or something completely different. So for example, other common things are you'll have samples that just didn't perform very well. The DNA quality wasn't great, or the RNA quality wasn't great, or somebody kicked the sequencing machine. That happens. Sometimes you'll see on a run and there's one base that's off, and it's off in every single individual. Like that literally does happen. Somebody kicks it. Or you just simply get failed reactions. As I said, you often get sample mixes. These are really common. I've seen somebody here, a Ph.D. student, working on a study where every single sample in their entire cohort was mixed. Like probably somebody had a whole row of them in a rack and got off by one, and literally every sample was not what he thought it was. So you can imagine what that would do to your experiment when you actually come to ask questions like, what's differentially expressed or whatever. Like, forget about it. Like, you need to know what sample is what. You often will get systematic technical biases that will pop up. So these are things like batch effects. Traditionally, people have always thought of microarrays as being prone to batch effects, whereas things like sequencing is digital and doesn't have that. That's complete rubbish. Sequencing has just as many batch effects as do microarrays. There may be just slightly different kind of batch effects, but they very much exist. And it's really important to understand what those are and how they might affect your analysis. Maybe it's only affecting certain samples. Maybe it's affecting, the sample that was in the corner of the plate or something. But again, you need to understand what that is to be able to appropriately account for it in your analysis, or maybe just exclude it. And then you can get other weird things going on, like cryptic relatedness between samples, or like I say, sometimes just odd stuff that you just don't expect. And so a really useful method to start to look at these kind of things is something called principal component analysis. I'm not going to teach you any of the practicalities about how you write scripts to do this. I don't know how to do that, but I can tell you all about how great things you can do with it. But it's basically a really useful tool for understanding your data, a way of just visualizing large-scale data, whether it's gene expression, methylation, SNP level, whatever, and getting insights into the patterns in your data. It's basically a mathematical technique that, probably the best way of explaining it is it takes this large, multi-dimensional data cloud and tries to draw vectors through it that explain the largest fraction of variance in that data set. So it'll produce principal components, which are ordered 1 through n. Principal component 1 always explains the largest amount of variance in your data set. Principal component 2, PC2, explains the second largest amount. And these are all completely anonymous things. You have no idea what PC1 is. It could be gender. It could be day of the week that the sample was run on. It could be how long your sample was sitting in the freezer. You don't know, but it's just some factor in the data that's affecting the results you're getting. And these are basically variables with a number, and you can plot these as a way to visualize what your data looks like. So this is an example of a PCA plot. The most common sort of PCA plot, usually people will plot them as a two-dimensional scatter plot. Sometimes people will plot three-dimensional ones, but they're kind of hard because your screen is always two-dimensional. And usually people will plot PC1, which is the vector that explains the largest fraction of variance, versus PC2. You can plot PC1 versus 3, PC2 versus 7, whatever you want. I'm going to walk you through some real-world examples. These are all data, actually not quite all, mostly data that's come through my lab. Some of it's data from other labs that I know about, but these are all real things, so none of this is made up. And what we're looking at here is genome-wide methylation data generated using an array platform that looks about 500,000 CpG sites across the genome. And the particular experiment that was done here was 48 samples were analyzed, and these 48 samples were derived from 16 individuals. And in each individual, a blood sample had been taken and sorted into 3 blood cell subtypes: CD4 positive, CD14 positive, and CD19 positive cells. So 3 different fractions of peripheral blood. And I can't remember what this was in the context in, maybe a study on MS or something, I think. I was helping a collaborator with. And like I say, the first thing we did before we even do any analysis, like the biological questions they have, is make one of these plots. And the reason why is you start to see things like this. So firstly, you can see most of the samples are up here, and then there's one guy here and one guy down here that are huge outliers. Just to explain, whenever you do a principal component analysis, it's really nice to know what fraction of the variance in your data each principal component explains. So in this case, like I say, we don't know what PC1 is. There are anonymous numbers ranging from -100 to plus, I don't know, 70 odd. But we know that this first principal component explains about 28% of the variability in our data set. So it's more than a quarter. The second one explains 21%. So these two variables are explaining nearly about half the variability in this entire data set. But important thing, therefore, to bear in mind is these scales are not the same. So this is explaining more variance than this one. But the main thing to notice is there are 2 strong outliers. This one's a minor outlier. This one's a really strong outlier. If we then take the same data and we represent it in just a more traditional format, so this is, for methylation, it's called an M value. It's just sort of basically the peaks over here are methylated sites, the peaks over here are unmethylated sites in the genome. You typically, in normal people, see this bimodal distribution, but you can see there's 2 samples here that have a very different profile, and not surprisingly, they are the 2 outliers. Now, it's theoretically possible there are 2 subtypes of cells in these individuals that have completely wacky methylation. The more parsimonious explanation is the experiment failed. So a conservative thing to do here would say, okay, these 2 data points are bad, let's throw them away. So if we then do that, throw out these 2 and then focus on the remaining 46 samples that are not obvious outliers from the main group, and that's always a qualitative decision. Remake the same PCA plot, but now I say after removing those 2 people, focusing on what we think are the 46 good quality samples, we now get 3 clear clusters in our group. Now PC1 explains 38%, PC2 is 12%. And what was done here by the bioinformatician that was working on this is they plotted, firstly, to see if there are systematic batch effects, because the way this platform works, it has an array slide, a microarray slide that has 12 different arrays on it. So basically, there are 4 slides that were run, each with 12 samples, and he plotted it with each of the 4 slides in a different color, just to see if there's anything systematic going on, with the different intra-experiment batches, you could say. And the first thing to see is there's no obvious effect there. There's all 4 colors represented in 3 clusters. So a reasonable conclusion would be there's no obvious effect due to the slide that each sample was run on. This is now exactly the same plot, exactly the same data, but now just recolored the points. And now what we've done is start to look into the biology of the experiment. So we've recolored the points based on the cell type. So I said there were 3 different cell types run in this experiment. Can anyone spot the problem? Everyone's really quiet. Yeah, okay, so this one sample here, I think the red and the blue were just the 2 different, I can't remember, the genders or slides they were on, but 47 should be clustered down here with the other dark blue guys, and it's not. So we went back to the people that ran the experiment, the technician, and said, hey, are you sure this one is, I can't remember, CD4 positive, whatever the groups represent. And they went back and looked in their notes and compared some things and came back and said, oh yeah, you're right, I think we made a mistake, that sample was mislabeled. So instantly, with a couple of plots, we've found 2 bad samples, throwing them out. We found a sample mix. We can correct that by hopefully figuring out where things should be, where the swap was, or simply by excluding it. And if you hadn't done this and you didn't know about that, you would probably A, have some flawed analysis because you have 2 really bad individuals in there, and 3, if you came to do anything where you're saying, oh, I'm going to compare this cell type versus that, that's going to be wrong too, because now you have cell types that are mixed up. So you can do the same thing for many other kinds of analysis. So I said, pretty much every large cohort study I've ever worked on, you always get sample mixes. This is just a really simple example of a test that I'd always recommend doing. It's just simply checking gender. So even if you know nothing else about a sample and you just want to say, hey, did anything get mixed up? We're simply checking each sample was annotated as being male or female. If we analyze the data in the right way, we can check, is every male sample, is the data consistent with them being male? Is every female sample being consistent with that sample being female? To do that, what we did is instead of plotting all the data in the genome, we simply took data points on the X chromosome. If any of you know much about epigenetics, females have something called X chromosome inactivation, which epigenetically modifies one of their 2 X's. Males don't have that. So you get a fundamental epigenetic difference between the 2 genders on the X chromosome. So that's what we're visualizing here. So presumably in this case, PC1 is the sites on the X that are being modified as a result of X inactivation. That's the main driver of it. We don't know for sure, but that's a pretty decent guess. And the obvious thing you can see is there are 2 samples that were labeled as female that cluster with the males, 1 sample that's called a male that clusters with the females. So again, we have at least 3 samples that are being mixed here. They're not what they're labeled as. An important thing to bear in mind here is you only ever go and spot with this kind of analysis half of your sample mixes. Because if a female is swapped with another female, it'll cluster in the right group, and same with male to male. You'll only see male to female and female to male switches. So probably there's another 3 sample mixes, you would guess, thrown in here, which we can't spot with this analysis. But you can do this with gene expression, you can do it with SNP data, most kind of things. It's just an important question of what do you put in. If we put in data from the whole genome, we probably wouldn't see, if gender wasn't the major driver of the entire genome, the X is only about 5% of the genome, we may not see any difference clearly based on gender. But by putting in data only from the X, then that's the main driver of our plot. You can also use principal component analysis for checking for batch effects. A batch effect is something you see all the time in genomic data. And basically, if you imagine, you have a bunch of samples that you're gathering over a course of 2 or 3 years as part of a big study, often what people will do is say, oh, I've got my first 100 samples, great, I want to get some data, let's send them off and analyze them, get them run at the core here. 6 months later, they get another 100 samples together, great, I'll send them either to the same core or to a different core, where maybe things are a little bit different. Probably the batches of the arrays that you bought from Illumina or whatever are now a different lot number. Maybe they updated the software on the machine, all these kinds of things happen. And so what that means is even though you may be using exactly the same platform, there are systematic biases that creep in and permeate your data depending on the batch. And like I say, that can be caused by lots of different things. I've seen it with DNA that was kept in a freezer or RNA that was kept in a freezer for a month, and it turned out you could see some effect happening at one particular date, and we went back and it turned out that person had moved labs and the freezer had been unplugged for a few hours, and probably that had affected their RNA quality. You could see that in the data. What we're plotting here is 1,500 individuals. Again, here this is profiled with these Illumina methylation arrays, but you could do the same thing with many other kinds of data. And here we've plotted, I think there's 9 different batches of data, all from all over the world. This is all publicly available data we downloaded from the web. And you can basically see that each batch has its own particular bias. Again, no idea what these principal components are, but you can see that the gray samples basically have 0 overlap with the yellow, have 0 overlap with the purple. Some of the other batches overlap to some extent. But it's telling you there are fundamental differences in your data that vary batch to batch. And so if you were to say, all right, I'm going to ask the question, what's different between my yellow batch versus my gray batch, because these samples have, diabetes and these don't, you have a bit of a problem because You don't know now if you find things that are different, is that different because these do have diabetes and these don't? Or is it different because these were ran in Germany 2 years ago and these were ran in the States 6 months ago? So again, these kind of plots can be really informative even before you start to ask any questions of your data, because it gives you a sense of what's reasonable to ask, what you might expect, you know, given your system. Feel free to stop and ask questions. It makes things more interesting. Yeah.
Is there any way to correct for the batch bias to like somehow get it all?
Yeah, I should have thrown some slides on this. Yeah, so there's normalization approaches that people use for many different kinds of data. So quantile normalization is one approach. Let me explain what quantile normalization does. I can draw on here. So imagine you have, let's say, sample 1 and sample 2, and then this is 0.1, 0.2, 0.3, 0.4. And we're going to give these values of, say, I don't know, 6, 4, 7, and 2. And in this sample, it's 5, 3, 8, and 1. What quantile normalization does is it's an approach to try and correct for things like this, where you maybe have 2 batches of data, or even a single batch of data where you just have sample-to-sample differences. So if we go back, I showed you this plot. These are all these samples derived from the same batch, and you can see, you know, these peaks don't align completely with each other. You know, the blue sample up here has a preponderance of methylated sites compared to this orange sample or something. So there's some subtle differences going on, which could be due to DNA quality, maybe one DNA labeled a bit better than another or something, or, you know, had slightly longer in the wash step or something, just due to the way the technician processed it. So what quantile normalization does is takes a large data distribution like this and basically forces things to look the same. It will force things to have an identical overall distribution. And the way in which it works is you basically, quantile normalization takes the top ranked value. So you first rank your values in each individual. So our highest ranked value in this person is 8. The highest ranked person, this one is 7. Happens to be the same probe or the same data point. And we average those two, so 7.5, and replace the value with the average. We then take the second ranked value, which again will be 6 and 5. We then replace those with the average of them. So those become 5.5, 4 and 3. Those become 3.5. And then, again, average these, that will become 1.5. And now what you can see is, I said it forces the data distributions to be identical. If you look now, the way I drew it, it will usually be different probes that are maybe the first ranked in sample A versus sample B. But you now see that basically the values are identical, or the distribution of values in the 2 columns is the same. So if we were to make a density plot of these 2 samples now, they would perfectly overlay each other. That's one kind of crude approach that you can do. There are various other softwares around that are often platform specific for expression, arrays, sequencing, epigenetics, things like that. There's a package called Combat. The BAT stands for batch, that people claim removes batch effects. The reality is they never fully remove batch effects. They will help, but don't expect them to work wonders. They will help with subtle things. If you have large scale differences, they're never going to be able to iron out. Yeah.
Are there advantages or disadvantages to using z-scores as your normalization process?
Yeah, so for anyone that doesn't know what a z-score is, again, you're basically superimposing your data onto a normal distribution and converting whatever your input value is to a number of standard deviations from the mean. So again, you force everything to have the same mean, and the same variance.
The data will assume to follow the normal distribution. So it's a parametric if they don't follow the normal.
Right.
Yeah, I was going to say, yeah, if you have a...
No, it's better, I think.
Yeah, you're forcing it to be normal. I don't know. I don't have a ton. If anyone else has.
The assumption it's normal, more or less.
Can you speak up? Sorry.
On the assumption that you have linear regression. So those unitless values are actually the eigenvalues of
Okay, so he knows better than me.
spread in your data. So the z-value is very less stringent to analyze or put your data in.
Well, no, so right, PCA is, as I understand, a test for outlier, right?
No. PCA is just a method to identify the main sources of variability in your data.
But right, so if you, as you noticed with your batch effects, the way that you corrected it wasn't with a PCA, right?
No.
So you're
You don't use...
with the quantiles, correct?
You can use quantile normalization is an approach that is applicable to some kinds of data
Right.
to basically force it to have overall the same distribution.
And so what I'm asking is, for z-score normalization, what are the advantages and disadvantages? So yeah, you have to, so I understand that you have to assume a normal distribution. You assume that if you know that your distribution is not normal, it's absolutely not a normalization method you should use. Are there other assumptions inherent in that, though, that are also detrimental to even z-scoring?
I don't know, is the honest answer.
Okay.
I haven't worked with it much. If anyone else has any thoughts. So, sorry, just before we go. One other thing with quantile normalization, because I didn't explain it that well, because you're basically, you would force these distributions to look the same, there's the potential with normalization approaches you can actually wash out real biological signal. So the underlying assumption with applying an approach like that, saying, you know, okay, I'm going to presume that the differences I see are largely technical in nature, and by forcing them together, I'm hoping to remove technical biases but retain my biological signal in my data that I can then analyze subsequently with some statistical test. But they can, you know, get rid of true biological signal too. The underlying presumption is that the overall distribution of data is fundamentally, on a genome-wide scale, the same in the samples that you're putting into that normalization. So for example, say you had data from sperm, which is typically very highly methylated, and some other tissue which is globally less methylated, you would not want to quantile normalize those two different cell types together, because now neither of them will represent the original. One was more highly methylated than the other globally, and now by normalizing, both will be halfway between what they originally were. So you would have imposed a whole global change in the genome methylation that you're getting out after normalization, which does not represent what is truly there. So that's just something to bear in mind. You know, or cancer. Some cancers get incredibly highly methylated. So you need to be very wary of that. And sometimes the simple answer is you don't know. It could be biological, it could be technical. And then ideally you'd want some points in the genome that you know are truth, and you can ask, okay, pre and post normalization, can I still see the signal I expect to be there? You know, for example, the X chromosome between males and females, you know, there's some true biological signal. You could ask things like that. So anyway, sorry, there was a question. No.
Just a follow-up comment. So you're saying some of these batch effects would seem to be different racial mixes between the centers that they...
It's possible. So yeah, it's possible. I mean, globally, if we took the same cell type, so CD4, you know, cells from a bunch of different people, we would not expect you to have more methylated overall CD4 cells than me, for example. I guess it's possible, but I think that's unlikely.
Okay, with the specific cell type.
Yeah, so in that case, but say we were to do quantile normalization, in this experiment, probably the appropriate thing to do would be separate out the three cell types through input and quantile normalize them separately. Might be the better way of doing it. And then, for example, I showed you data on the X chromosome. You know, this is just the PCA plot, but if you looked at the density plot of the X chromosome, like this is taking all this is actually only based on chromosome 1 through 22, the autosomes, the density plots look fundamentally different in males versus females on the X. Females have this kind of three-humped peak with a bunch of intermediate methylation values, males don't. So quantile normalization, if you have different genders, you always want to split your gender, quantile normalize the X separately as you do to all the other chromosomes, because we know biologically there's a huge difference in methylation or gene expression on the X versus the autosomes. So there's always, you know, you need to understand the biology of your system not to do stupid things informatically. And then there's other things. So this was another plot that somebody in my lab made. Again, we were taking a whole bunch of public methylation data. They made this principal component analysis plot, you usually don't see ones like this where there's this massive difference. We don't have the variance of PC1. What I actually realized later on, this was a student that was coming in, was kind of new to dealing with this kind of data, and we figured out later on actually this was one particular study they downloaded it, and they didn't realize this one was sorted separately. When they downloaded it from the online database, these probes were sorted by, not by chromosome and position, just by ordinal number of the probes, which is some anonymous ID. And they didn't realize that, made the plot, and came out completely different. We ended up throwing this away originally, but it was a stupid mistake on the part of the analyst. But again, it's highlighting something like, huh, that's weird. You know, it gives you a mental check just to go back and make sure I didn't screw something up. This is another example of a study we worked on. This was RNA-seq data, and so the input to this was not methylation values per CpG, but it was, I think, normalized RPKM, so read counts or something. And this was the PCA plot we got. The experiment was, this was some rat brain data. We had some rats that had been treated, some rats that were untreated. And when we just color the points by treated and untreated, it's clear you have two distinct clusters, but there's no obvious grouping of treatment with cluster A or cluster B. But then when we add on other data that we have, so obviously we knew gender and things like that, it's clear then that there was a huge difference between the males and the females that were treated. Again, you might not necessarily expect that. We didn't think that there would be enormous fundamental genome-wide differences in gene expression based on gender. We know there are some genes that do have that, but not on such a big global scale. And so again, we went back to the people that had prepped the samples, asked them, said, hey, like, were you expecting this? And it turns out they told us, like, oh yeah, we did all the males in 1 week and sent them to the core, and then we did all the females the next week and sent them to the core again. And then the core had decided that the preps they got with the females, the second weren't so good, so they put them through an extra purification step when they made the RNA-seq libraries, and there's your result. So basically we had this enormous technical confounder in that the two batches that happened to correlate with gender had fundamentally very, very different data, basically because of the library prep protocol that had been used to create them. And the really unfortunate thing was, one of the analysis that people wanted to do was ask, are there gender differences in the treatment they're working in? The answer is, yes, there are gender differences, but I can bet you that it's nothing to do with gender. It's just to do with the fact that you didn't prep your samples in a very good way. So I'd say the lesson here is you always need to be very wary about these kind of things. If ever you're working with people that are preparing samples to go and be sent to some core, it doesn't matter whether it's sequencing or arrays or whatever, always think about what's my biological question, what are my variables, and kind of distribute things in a way that is where you disperse those variables evenly across batches, across slides, across plates, whatever it is, like that. Yeah.
At what level do you say, oh, I can fix it with more addition, or I should just go in?
I would say the naive person would say, hey, we'll normalize it and it will be great. For me, having worked on this for 18 years, I would say, oh crap, we just wasted $20,000. But the people who paid for it don't want to hear that, so they kind of say, no, keep going, it'll work. And you're like, probably not. So yeah, this was a study that, in my view, was not very successful, but was basically killed from the start because the way the samples were prepped and processed was not done in a good way. I actually showed you this kind of similar slide yesterday. So you can use PCA to do other stuff, not just tell you about batch effects and sample mixes and screw-ups in data. But this is a PCA plot of SNP genotype data from different European populations. And here you can see the really cool thing of it clusters individuals really, really well based on their geographical region where they come from. So here we're looking at real biology. I don't think it's anything to do with how the arrays were run or anything like that, but it's showing you ethnic differences, or real biological differences in allele frequencies within different populations. I mentioned at the beginning, and all the plots I've shown you so far have been plotting PC1 versus PC2, the two principal components that explain most of the variance in data. You can make plots with other ways, so I'll show you in a second, like PC1 versus PC3. This is similar to that plot I just showed you here is European population SNP data. This is SNP data from the 1,000 Genomes project, the various different, some of the subgroupings that went into that. So here, if we just look, this plot is PC1 versus PC2. On this plot, we've basically got Chinese from Beijing, I think these are Chinese from Denver in the US, and Japanese from Tokyo. They basically all overlay each other perfectly. If we then change that and now plot PC1 versus PC3, suddenly the Japanese now cluster with Tuscans, so people from Italy, whereas previously the Tuscans were down here, so very separate. So again, we don't know what these vectors are. You know, it doesn't represent, in this case, world geography. I don't know if we could do that, but just to show you that, you know, you can plot different principal components and they will separate your data in different ways. We don't know what those things are, but it's there. And then this is just a random thing I grabbed from the internet. This is some guy that runs some bioinformatics blog. He's from somewhere in South Asia. And I think he got him and his parents done with 23andMe, got the raw data from 23andMe, and then decided to see what his ancestry was. And here, so we made a similar kind of PCA plot with a bunch of other individuals he downloaded from other sources. And here's this guy's parents, where they are. If you kind of squint a bit, you can see this is Middle East. This is East Asia, so it's kind of China, Japan. These are people from sort of Mongolia, Pakistan, and South India. So you can almost imagine his India sticking out into the Indian Ocean. Over here's China. So, and this actually fits perfectly where he's from, sort of the Chennai region on east coast of India. So just a neat little thing. I talked about genome-wide association studies, where you're comparing, say, allele frequencies, looking for differences in maybe case versus control groups. That's a popular method. One of the weaknesses of GWAS is it can be very sensitive to population stratification. And what I mean by that is, say, for example, you have 1,000 cases you've got from New York, which has a very diverse population, people derived from all over the world, and 1,000 controls from, let's say, LA, where the racial ethnic mix of those individuals is going to be very different. You're going to get different allele frequencies that's a result of your sampling, not maybe due to your phenotype you're trying to study. And so often people will use PCA just as a way of recognizing, just as I showed you that earlier, that basically the ethnic background of your individuals. So here's another of these PCA plots, just plotting, there's maybe like 2,000 or 3,000 individuals here. And you might decide, okay, I've got this obvious cloud of people going over here. I don't know what's different, but clearly their SNP genotypes are saying they're fundamentally different to my main grouping here. You have some less obvious cloud. These principal components all have values. You may decide, hey, I'm going to cut and exclude all these people over here because they're more divergent than my main population. It's a very arbitrary, sort of qualitative thing. There's nothing magic about 0.25 as a value, but it seems to be a reasonable place that would separate your main cluster from the rest. And you might also want to say, oh, I can cut here. It doesn't make a lot of difference. There's only one person. But a really important thing to think about is how much variance do these two principal components explain. Here it's 28%, here it's 5%. So this axis actually explains, about 5 or 6 times more of the variability in my data than does this axis. So in fact, maybe a better way of thinking about this plot would be that. So even though R plots it in that format, this is actually scaled based on the amount of variance explained by each of these principal components. So when you think about it in that way, this one is 5 or 6 times more important than that. Yes, you probably would want to cut about there and get rid of these guys. And maybe cutting here is not so important. Just something to consider. So another sort of very complementary approach to PCA is something called hierarchical clustering. This is another methodology that you can use to measure similarity or difference between samples. And what it does, unlike PCA, is basically visually clusters or directly clusters things together that are more similar to each other. And your results then are plotted as what's called a dendrogram, or a tree, if you like. You think of this as an inverted tree with the branches and the twigs. It comes in 2 flavors. It can be unsupervised, where you don't tell the algorithm anything about your data. You don't tell it, hey, these are males and these are females. Try and cluster them based on that. You just say, here's the data. Off you go. Or supervised, where you pre-specify groups and it will try and force things into those. And just as we saw on a PCA plot, you have the principal components that have values attached to them. In these clustering trees, these dendrograms, the important thing is the length of these branches. So you see, you know, this branch is very short, this branch is relatively long. The relative length of these branches is a quantitative value that tells you the extent of similarity or difference between samples. So basically it's saying the difference between sample 3 versus sample 4 and the 5 is relatively small versus the difference between 1 and 2 versus these ones, because this branch is, you know, maybe 8 times bigger than that branch. And so it gives you a very sort of complementary approach to PCA. This is the exact same data plotted in PCA format and in an unsupervised cluster dendrogram. Here you can clearly see that all these red and yellow guys, which are right here, cluster separately to the blue and the green. So, you know, that may be a conclusion that you wouldn't have gotten from the PCA plot. So like I said, it's plotting the data in both ways is often a nice way of doing it. And the nice thing here about this clustering dendrogram, you probably can't read because it's really small. This was actually a bunch of samples we ran in duplicate. So we had the same single biological sample that was split into 2 and processed in parallel. And the clustering dendrogram nearly always puts the pairs of samples together, which is a kind of a nice, just, at least in this case, sort of, you know, reassuring thing that your data is hopefully fairly good. The replicates cluster together as they should. And people actually use things like this. So a technique that has been around for quite a while now here, for example, taking gene expression data from breast cancers and using PCA or clustering to basically identify different subtypes of cancer based on their gene expression profiles. So you can use it for things like that too. And it turns out this actually characterizes breast cancer into different forms that maybe have different treatment protocols, things like that. I already mentioned things like batch effects. I just want to throw this one up to show you. This is a paper that was published a couple of years ago in some relatively minor journal, but it just really nicely illustrates false conclusions people can come to, let's say. So the title of this paper is Stable Ethnic Variations in DNA Methylation of Human Skin, okay? And what they're showing you is a dendrogram that has African individuals, European individuals, and Asian individuals. And based on this dendrogram, they said, well, look, the Asians cluster separately to the Europeans that, apart from these being different to the Africans. So therefore, that's our conclusion, okay? If you then add on the batches that they ran their arrays in, you might come to a very different conclusion. I would say probably a more appropriate conclusion for this paper is batch effects really, really influence my data heavily and can make me have very wrong conclusions. So just as a warning to you guys, like, this is why I'm saying these things are really important. There's a lot of people out there that don't appreciate the importance of doing quality control, systematically mixing your samples. If I was running this experiment, I would probably run them, it's not that many, run them all as a single batch, not as 4. And if you had to separate them into batches, run some Africans, some Asians, some Europeans together in each batch, you know, so that you wouldn't be misled in this way, let's just say. Any questions or more thoughts? No? Anyone sitting there worried about their own data now? Yes, everybody is. Yeah, what?
So, how did, just curious, how did people find out about this? Are they required to publish the batches?
I don't know. This actually came from a friend of mine. I don't know how they saw that, whether the data's available or whether it's said in the paper somewhere. I don't know. But I mean, the examples I showed you of that rat RNA-seq where we went back to the text and they said, oh yeah, the males and the females were run differently at different times with different protocols. Like, that's a pretty bad example of it. But yeah, the bottom line is, like, in genomics things, it's nice to be involved right from the start. And even if you're an informatician just working on the analysis side, like, get together with the people who are running the experiment, make sure, I mean, the ideal one is you have all your samples together and then you run them all at the same time. Even then, you know, even if it's sequencing and stuff, things still have to be batched up in some way, in a 96-well plate format, you know, or 16 samples per array or something. look at all your variables, age, gender, disease status, you know, and randomly, or not randomly, semi-randomly assign them across whatever batching has to be done. So if you have 96 and half your samples are female, half male, put 48 males and 48 females on that plate, but not 48 over here and 48 over there. Mix them, so it's male, female, male, female, male, female, that kind of thing. That's what we do. And it will that's by far the better way. I mean, somebody asked me about normalization. 1,000 times better to get involved right at the beginning of the planning, even from like the extraction stage, and how the data is generated, and make sure it's done in the proper way, rather than have to try and worry about it afterwards and think, oh, can I apply some normalization algorithm that's suddenly going to make everything better? Because it won't.
So when trying to decide whether things are parceling out according to some sort of group you have in mind, besides just eyeballing it and maybe drawing a convex hole around it, what tests are available to actually test whether those statistically significant different groups?
I don't know of any particularly. I don't know if anyone else does. And for me, it's usually just a qualitative decision. You kind of have to decide how far of an outlier is too far. Sometimes we kind of keep things in, but just flag things visually of like, oh, that guy was like on the edge, you know, by PCA.
Overlap between two groups, because eventually, you know.
Yeah, but the problem is sometimes, like I say, usually you're looking for some biological difference between samples, so it could be that if you see separation, that may be real. You always have to think about these things in the context of what's your system. Maybe you're studying, let's say, Alzheimer's disease, of people with Alzheimer's who have died of it and people that haven't. Like, their brains are fundamentally different. Like, they decay and, you know, lose a third of their volume. Whereas if you're looking in there, you might expect a really strong biological difference. If you're, some people are doing studies of anxiety and they're testing something in blood, there you're not going to fundamentally expect that the people that are anxious have a fundamentally different profile to the people that are not anxious. Like, you're looking for subtle things. So yeah, I always think about these things in the context of what's the biological system. Am I expecting to see biological or technical effects that are of similar magnitude or very different magnitudes? Or and again, it's always nice to have known things in there. So like, you know, always a simple one is gender differences, male versus female, looking at the X chromosome for things, or other positive controls if you can put into your assay things that you know are already different that you can ask, you know, pre and post normalization, or in these different batches, is that still there? Things like that. So no, I don't know of any hard and fast ways. It's more kind of gut feelings and just understanding your system, I'd say, and putting in some good controls if you can. Yeah.
Two questions. One, like in the PCA, is over here the y-axis some factor that you're unaware of, or are you choosing how to cluster these?
So this is unsupervised clustering. Yeah, it's kind of analogous to PCM that's measuring global difference. It's a linear scale. So yeah, like I said, the length of these, of the y-axis is telling you you don't know what the factor is that it's measuring of difference, but it's a linear factor saying, you know, if this branch is, say, 10 times as big as this branch, that's saying this sample is 10 times more different than to these than that one is to those, if that makes sense.
My second question, how does this give you any more information than the PCA analysis? You say it's complementary, but what does this provide that?
Well, you might see things here. So from this PCA, I mean, you can see these blue ones cluster and these green ones cluster, but you wouldn't you wouldn't necessarily say that where are we? These green ones are more similar to the blue than the blue are to the red and yellow. Whereas in the cluster dendrogram, it's clearly saying, yeah, the blue and the green are one cluster, and they're very different to the red and the yellow. And it's also giving you like the pairs of saying, yeah, these two are much more similar to each other than they are to these ones over here. So it's just a kind of different way of looking at it. I don't know if anyone else has another perspective, but sometimes it will tell you exactly the same thing. Sometimes it gives you some kind of complementary information.
How pervasive do you feel like some of these batch effects are in the data that you see every day published?
Yeah, a lot. Where are we? This plot's a pretty good example. So this is a platform that tens of thousands of samples have been run on, this Illumina 450K methylation array, and this is just 9 different studies. That's the data. So it's just telling you if you took a batch of samples and ran them 9 different times in different places that, you know, sometimes the data is going to, you know, if you see there's 2s and 3s mixed in here, they cluster very well with each other, but they're totally separate to the 7s or the 9s. So
how often do you feel like you see published data that you're like, yeah, that's probably not real, it's probably batch effect?
quite a lot. Yeah. Yeah, I'm always reading between the lines of seeing what people did or what things didn't replicate, or but it's not the kind of stuff that people usually tell you about. Like, they won't say, I mean, sometimes some people will say, yes, we had all the samples are processed in a single batch and we did this. Other times it's just usually we sequenced them. And it's not until you get the data and dig into it that you can see, oh, yeah, like your cases and controls are really different, or, you know, a third of your samples look totally different to the others, even though there's no reason for that. Probably they're run in a different batch. So yeah, it's a really important issue. Yeah?
Also heard about k-means clustering. How does that compare to PCA or hierarchical?
I'll be honest, I've heard of it too, but don't know much about it. My understanding is it's a technique where kind of like dendrograms, it tries to group things together based on some similarity, but that's about all I know. I don't know, some maybe you know more.
You have, by saying k-mean, you have to give you have a expected clusters, which means you have some experience about your data, and try to approximate it. That's kind of supervised.
Yeah, I don't know if that's helpful. But I'm not a bioinformatician, I just do a lot of genomics. So I just hire the bioinformatics people to make all this stuff work. All right, where did we get to? Oh, here. Yeah, and I just wanted to give you guys a few examples. This is just kind of random things thrown in, but from my experience, one of the big challenges or problems with genomic data is usually you can end up with millions or even billions of data points. You know, you run a sequencing run with a high seq, you get 200 million reads. You know, you run one of these Illumina methylation arrays, that's 500,000 data points. You know, we're doing studies with like thousands of individuals, so we literally have billions of data points. Like, how on earth do you make sense of that? You can't sit there and stare at a table. Sometimes you can, but unless you're looking for something very specific, that's not going to mean anything to you. So for me, ways of visualizing data is a really key thing. Making different kinds of plots, because you can start to get some really important insights into maybe biological or technical trends that are going on in your data that you may really want to know. And so all these PCA plots and things I've been showing you is one way of doing that, but there are lots of other things. So here's just a few examples. The backstory to this was we were working with some mouse with a knockout of a particular gene, the folate receptor 1 gene. And these mice were supposedly inbred, like, you know, 20 or 25 generations, so supposedly completely pure background. And we had a bunch of results where we had clustering of these genes that were differentially expressed in a certain region of the chromosome, and it's like, oh, that's kind of weird. And so here my bioinformatician just made a plot. This was actually calling SNPs in RNA-seq reads and then mapping where they are versus the C57 black 6 mouse reference genome onto which this knockout was supposedly bred. And basically you can see that when we just plot the location of these SNP variations in this inbred mouse, of which in theory there should be almost none, you can see there's a really obvious cluster right here, and it turns out that's exactly where the folate receptor 1 gene is. And when you think about it, that makes perfect sense, because if we have a mouse with a knockout, every generation it's being back-crossed to try and make it purebred. You always, or the breeders are always keeping the mouse with the knockout, because that's what we want to keep. And so while all the rest of the genome will melt away and be the background strain that you're back-crossing it to, the black 6, you're always going to retain the area of the genome with the mutation in, otherwise you don't have a knockout mouse anymore, you have a normal mouse. And so the one bit of the genome that is not on the black 6 background is exactly where the knockout was, if that makes sense. So anyway, it just kind of helped us like understand what's going on in our data and decide, oh yeah, all these places, these genes that are showing differences in expression is probably just because They're genetically different. They're on a different mouse strain than the rest of the genome. Yeah.
What do you use to create that plot?
Again, I'm not a bioinformatician, probably done in R, I'm guessing. I've never written a line of code in my life, so. Here's some really other neat examples of just data visualization and what it can tell you. So I talked about this paper yesterday briefly. That was Jim Watson's genome that was sequenced with 454 sequencing. So that was the first diploid genome that was sequenced. And this is just, I think, like supplementary figure 1 from their paper or something. It's a plot of read depth across the genome. And if you're sequencing a genome, so you take Jim Watson's DNA, you shear it into fragments, you throw it into either some vector or ligate adapters on, and then you throw it into your sequencing machine. What you're hoping is you're pulling reads randomly from across the genome. And so you're looking for a plot that basically has a very smooth line where your read depth is, nicely centered with no particular skew one way or the other, suggesting you had a preference to sequence GC-rich regions or not, or whatever. So the odd thing for me in this plot is this little bump here. Like, why is there an extra little bump right there? Often when you make plots like this, there's literally probably like hundreds of millions of data points going into this plot. It's the plot of fold coverage across the genome versus frequency of reads. Why would you suddenly get a little shoulder on it? So can anyone think? And I'll give you a hint, Jim Watson's not a woman. So basically, he has 22 pairs of autosomes, and the mean coverage on those is about 8x. And then he has an X and a Y that are only present in one copy. So all the other chromosomes are diploid. X and Y are haploid. So basically, this bump here is exactly half the size. There's 8x coverage of the diploid chromosomes, 4x coverage of the haploid chromosomes. So this bump here basically reflects the reads mapping to the X and Y in terms of coverage. So again, a simple plot like this, but it's benign. He's a man and he's got one X and one Y and not two of each. But it gives you some fundamental insights just into the biology and the experiment, and it makes sense. This is another plot from that paper where it does tell you something real about biology that you may not have expected. So this is a plot of deletions that were called, so taking these reads, and I think it was just looking at split reads, so where does a read map with something missing or extra added in? The x-axis is the size of the deletion that was made, and this is just frequency. And again, you see this kind of curve nicely down, but suddenly there's this hump right there. And again, when you get humps in plots like this with like many, many, many data points, it's usually telling you there's something going on. It could be technical, it could be biological, but it's saying like, what is this? And in this case, the size of that hump is about 320 base pairs, which corresponds exactly the size of a full-length ALU element. And so what we're seeing there is a preponderance of deletions or insertions in the genome that are presumably ALU elements that are polymorphic, jumping in, jumping out. And so there's an excess of structural variance of exactly that size, because that's the size of a full-length ALU. When it jumps in, it creates a 320 base pair insertion. So there you are getting some real biology. These are just some examples that usually when I get a big data set, we'll do some just preliminary QC, we'll do some analysis, generate some raw statistics, whatever, and then I'll usually just sit down and start making plots, sorting things by different ways, and often you'll stumble across things like this. Sometimes you expected them in data, sometimes you didn't at all, and you can often latch on to some interesting biology in your data set just by plotting, visualizing it in different formats. This is another one. This is something completely different. It's RNA-seq data from human brain, and this here we're plotting 600 individuals along the x-axis, and the y-axis is the taking transcribed SNPs and asking which SNPs are monoallelically expressed. So we know the individual is heterozygous at the DNA level. We take RNA-seq reads, map them to the genome, and ask at that heterozygous SNP, are you only expressing one or other allele or both alleles? This is the count of number of sites that only have one of the two alleles being expressed. And basically you can see there's one really, really strong outlier individual, another one who's popping up above the rest. And that's not biologically expected. Why should there be one individual that has 10 or 20 times as many monoallelically expressed sites as anyone else? Could be real. Maybe they have monosomy for a chromosome, but then they'd probably be dead. It turns out these two individuals actually have very low RIN scores, RNA quality, RNA integrity, and so probably it's a technical artifact. So it's just highlighting, again, 2 samples that had passed all other QC metrics that people standardly applied. But when you get a metric like this that's maybe more sensitive to subtle biases in the data, you can spot that suddenly, yeah, the RNA quality is having a big effect on this particular parameter in my data, and probably I should chuck those 2 out because it's probably not real. This is one other example. I think it's the last. This one is something we're just writing up now. It's methylation data across a population of fibroblast cells. And we took sites in the genome that show variation in epigenetics, sorted them by genomic position. So this is chromosome number. So top of chromosome 1 down to the end of chromosome 22. And what we're plotting here are the pairwise correlation values across the population of variation in DNA methylation. So where you have blue, that's saying a site here is very strongly positively correlated with a site somewhere else, either on the same chromosome or on a different chromosome. Red is strongly negatively correlated. So you can see there's, if there was, basically what this is telling us is there are places, on one chromosome that are, for some reason, really strongly correlated with the epigenetics on another chromosome, which is kind of unexpected. If you then add on Hox gene loci, so these are genes involved in just fundamentally controlling cell growth, what you can see is all these main clusters are the 4 Hox genes A, B, C, and D clusters that, for whatever reason, have very strongly correlated epigenetics in trans. And so it's basically highlighting trans-chromosomal epigenetic regulation going on, which you would have otherwise probably no chance of spotting unless you make a plot like this. And basically it turns out there are transcription factors that bind to all these loci and coordinate epigenetics in a trans-chromosomal fashion. So plotting you can also use just to get insight into your data and spot things like false positives. So again, this is a study we did. We were here correlating tandem repeats, and as they expand and contract, what that's doing to local DNA methylation. So we have a tandem repeat and asking for methylation sites around it. Each one of these points is an individual CpG. Which ones are correlated with the length of the tandem repeat in the population. And so it looks like we have a spike here with one particular methylation site that's reasonably well correlated with the size of the tandem repeat. This is at least just when we look at the R value. If we then actually plot the raw data, this is what it looks like. So this is tandem repeat size, and that's methylation level. There's nothing going on in the data. Basically what we just found here is some completely artifactual correlation. Everybody basically has 0 methylation. It just happens there's a tiny bit of probably technical variance that by chance just happens to correlate in our population. So you look at it on a global level that you might get out of a program, it tells you a p-value and r-value, you go, oh look, that's cool. You look at the real underlying data and it's totally bogus. This is another one. Again, you go, oh yeah, look at that significant association. This is the real underlying data. It turns out, in fact, most people have exactly the same allele. There's a couple of rare outliers, but really only a couple. There's 2 here and 2 there, and the blue population, 1 there and 1 there. And again, this is one I would not trust, even though the p-value on paper is good, survives FDR correction or whatever. It's really just driven by 2 outlier individuals. So again, some spurious correlation that, looking at the summary statistics, you would never see. And then after having looked at a bunch of these, you can come up with some additional filters, some metrics that you would say, all right, now I'll only trust sites that have some level of variation or whatever, and you'll get much better results overall. So again, plotting data is really key to understanding what's going on in it and giving you some robust results at the end. All right, we'll just start on this, and then I'll explain this, and we'll have a break in a minute. All right, so probably lots of you have read papers in Cell, I think loves to do this, where you will see maybe a plot like this of, I don't know, gene expression or mouse, whatever, BMI or something, and they'll have these nice bars with little error bars on the top and everything looks great, okay? These are terrible, really, really awful plots, and I will show you why they're so bad. So has anybody heard of box plots, violin plots, bean plots? Anybody not ever heard of them? All right. I'm going to show you why these are terrible ways of plotting data and why these are much better ways. And then we're just going to dig into this little tool here that's really handy to know. Okay, so I think up until now everything I've shown you has been real data. This is fake data, but I've done it just to illustrate the point. So here we have 2 data points. Let's pretend it's gene expression in some mice or something. This is the typical kind of plot you might see in Cell. And you look at that and say, yep, no significant difference at all. You know, the blues and the red mice are the same. There's a little bit more variance here, but you know, whatever, it's fine. Just as an aside, everyone always likes to plot standard error because that's your standard deviation divided by the mean and it's smaller. And it makes it look like your data is way better than it really is. It's true. So what I'm plotting now on these new plots, exact same data. This is in your Cell bar plot format with standard error bars. This is that same data now plotted in a box plot format. So here everything looks hunky-dory, no difference, everyone's happy. We plot the same data now in a box plot. It by no means tells you the full story, but you can see now suddenly that this standard error bar looks pretty good over here. Actually, you have massive variation in this left-hand side. You know, some people have values of 2, other people 14. The medians are still basically the same, but you suddenly get a little more truth as to the real distribution in the data. And do people know with box plots, this central dark line is usually the median of the population. The upper and lower extent of the box is usually the first and third quartile. So that's 50% of the data within there. The whiskers is usually, it varies 1.5 times the interquartile range, and then you'll plot individual outliers. But it just gives you a much better sense of the variation in the data, much better than this silly little thing. Same data now, we're going to plot it in violin plot format. Now you get a little bit more going on. What a violin plot does is not only has this same box that's basically giving you very much the same information as here. There's the median, the interquartile range, and the whiskers giving you the outliers. But superimposed on that is a sort of density plot format telling you that the fact that it curves in here, saying there's less data in the middle than there are at these 2 extremes. You still don't get the full picture until I plot you a bean plot. And that's actually the real data. So a bean plot is similar to a violin plot, but instead of having this box, we'll plot the individual data points as these, I don't know how well you can see them, these white lines. So what we showed up here is 2 things that look great, a slightly more variability in the left-hand group. That's actually the real underlying data. The conclusion you would get from these was there is no difference in my 2 groups. The reality is that this group is mutually exclusive with that one. This one has a bimodal distribution with no data points at all in the middle, which is where all the data is over here. It's an extreme example, but you can now hopefully see why bar plots for plotting population scale data are really terrible. And I have no idea why journals like Cell still allow them. And again, probably people are going to go home tonight and change their bar plots on their latest paper or whatever. I hope so. So what I briefly highlighted here, all these plots I showed you, there's this really neat little online tool called boxplot, tireslab.com. It has embedded R code, which you can just paste in your data, and it will make any of these plots that you want. You can also, if you prefer doing it on the command line, download it. And again, it has an R script that will do all this stuff. So I thought we could just have a little break now. If you guys want to wander off, get some fresh air, that's fine. Or I'd suggest go to this website here and just play around. Now we're going to move on. I think I've only got another half hour or so, so as I said, you guys will get an early finish tonight. We're going to talk a little about statistical power and why that's an important concept to understand. And another way of phrasing that would be, how big of a study do I need to do to be able to see what I want to see? Which, when you're writing things like grants, is a really good question to have an answer to. Otherwise, somebody's going to criticize you for that. This is a plot that's actually not too hard to make, that basically shows how much power do I have to detect a quantitative trait. So imagine you're doing like a GWAS study, and you're screening the genome, and you want to detect a SNP that's associated with, height, a quantitative trait. How much power do you have to detect that which we typically measure as, sorry, no, I'll start again. How much power do I have to detect that based on differing sample sizes? And so here we've plotted sample size of 50, 100, 200, and 400. And the way in which we, or the most important thing to consider in these kinds of analysis is how much of the variance is explained, how much of the variance in my data is explained by the thing I'm detecting. So say we're talking about body height, you know, maybe I'm looking for a marker that's going to explain 20% of the variation in height in a population. That's actually a pretty big effect. If you remember yesterday when we were talking about GWAS, a lot of the GWAS hits have, you know, R-squareds of like 0.05 or odds ratios of like 1.05. So in other words, they explain 5% of the variance. So this is like a modest to large effect size. And these curves are basically saying, okay, I'm trying to detect a variant that or something that explains 20% of the variation in my trait. If I am only looking at 50 individuals, I'm straight out of luck. You're never going to see it. Even if it's there in your data, like, don't even bother doing the study because you're not going to find it. If I now double my sample size and go up to 100 people, I have about a 40% chance of detecting that association. If I go up to 200, I'm solid ground. You're going to have about a 95% chance of seeing a variant that influences 20% of the variance in that trait if I use 200 people. The major thing that's driving these lines is, you know, what's my p-value to say I detect it or not. In a GWAS, we're looking at, you know, all the SNPs in the genome, so you have a massive multiple testing correction. This would be based on, I can't remember what, maybe it was 0.05 or something else. You know, that would be I was testing one SNP and I wanted to see it if it was 0.05 at nominal significance. But basically, plots like this are A, really good for grants, but B, I would say really important to understand the underlying concept when you're reading other people's papers, because again, it can fundamentally influence your conclusions. So say, for example, you know, sometimes I read papers and people say there is no evidence that there are variants that influence this trait or something. If they'd only looked at 50 people, that's a really dumb conclusion. The real conclusion would be, my study was too small to see anything. Whereas they may write in their conclusions, there are no variants that, you know, have a modest effect on something. So in other words, when you start to understand power of a study, you can start to interpret other people's findings, I think, in a more robust way. And vice versa, if you're going to go out and do a study yourself, and you estimate that, you know, maybe something you're looking for has this effect size, you would really want to be on this blue line, and you know that you now need to study 200 people to be able to see that. So if you can only afford to do 50 people, don't bother doing a study, basically. And if you only study 100 and you don't see something, well, probably your sample still wasn't big enough. It could well be there. Yeah?
How did you make that point?
I'll show you in a second. So yeah, so that was all done using this. Here's the web link. Sean Purcell actually used to be here at Sana. He recently left. This is a tool he made. It's called the Genetic Power Calculator. I find it a little bit cryptic to use. I was wondering whether to just go through and do a practical demonstration, but it's a little cryptic to use because there are all these terms where you have to dig down into the rest of the web page and read what they are. You know, like you have to, and many assumptions. That's one big caveat I would say about power calculations. Like the plot I showed you here, like it assumes that what your p-value threshold is, but also assumes, you know, are you genotyping your variant perfectly? Is there noise in my data? Lots of things that you really have no answer to what the reality is. So there are lots of presumptions thrown in. And so various ones of these numbers here, you just kind of have to put in 1 or 0 or have some guess. Like what's the case to control ratio for some diseases? You have no idea. Sorry, that one you do. Here, dominance additive QTL effects. Like for most diseases, it's really impossible to estimate what the truth for some of these is. You don't know. But basically what you do is you put in, once you assume some of these things, how many cases have I tested? If this was there's various flavors of this website, you can do different versions. What's my ratio of cases to controls? What's the p-value that I'm going to say is significant? Here it's nominal significance, 0.05. And then you put in you know, what power you want to be able to, what likelihood do you want to have of seeing that trait. So here is 80%, and then it will return to you how many samples you need to do. So it's almost like reverse engineering. You put in a different power, it will tell you how many samples, and you can create a matrix that you can then make this plot from, if that makes sense. But there are lots of assumptions in these things that, you know, it's kind of like a rough guide, but I wouldn't hang my hat on it. But, you know, at least you start to know, okay, I need to be up in the low hundreds in this kind of study to have a decent chance of seeing things. And they look good in grant applications. So like I said, once you start to understand these things and realize that power has a really strong impact on the validity of conclusions, you can start to interpret findings in other people's studies in a much more informed way. So for example, here's a paper in Nature Genetics from a few years ago. This was in some of the early days after the HapMap, where people started to look for genetic variants, so SNPs within our genomes that had significant effects on modifying gene expression. If you have a particular variant, does the expression level of a gene go up or down nearby? So what's called an EQTL study, Expression Quantitative Trait Locker study. And this particular study used 60 individuals, so it's pretty small, particularly by today's standards. And this is one of their figures showing the number of SNPs that influence gene expression, so the number of EQTLs they detected versus the R-squared value of those SNPs. So in other words, when you get to R-squared of 0.9, that means 90% of the variability in the expression level of this gene is dictated by whether you have this SNP in reference or alternate allele. When you get down here, it's saying, you know, only 15% of the variability in the gene expression is linked with that SNP. And basically what you see is there are very few variants with very strong effects, which we might expect. And then as you go to weaker and weaker effects, the number of variants they detect gets higher and higher and higher. And then it gets to this point, and then it suddenly falls off a cliff. Now, it does not mean that there are no variants in the genome that have weak effects on gene expression. In fact, probably this line goes up and up and up and up and up. And a more parsimonious, correct explanation would be when they get to trying to identify SNPs with relatively modest effects on gene expression, looking at 60 individuals, they just can't see them anymore. And if we go back to our power calculation here, we don't have 60, we have 50. Yeah, you can see as this line gets down to 0.3, suddenly they have 0 power. And so that's exactly what's going on here. With that sample size, they can only basically detect variants that have a strong effect on gene expression. Things with modest to weak effects, they can't see. I just explained this. You can start to calculate some of those things yourself. This is just another example of how to think about designing robust studies. Again, if you're writing grants or designing a study, like let's pose a question and, you know, figure out how many individuals might I need to look at to see the thing I want to see. So it's a pretty simple question we're going to start with. If I want to have a 95% probability, so almost certain that I'm going to see a variant that occurs in 1% of people, how many individuals do I need to study to see that variant? Okay, so let's say we're studying a population of people with, you know, congenital heart disease, and you want to find a gene that when mutated accounts for 1% of congenital heart disease cases. That's your question. Or more simply, I'm just trying to find an allele of 1% frequency in the population. How many people do I need to look at to be sure of seeing it? And it's actually not too hard to solve that problem just using some math. It's always good to have a handle of math and statistics when you're in the genomics world. So let's think about that. So if we start out with just looking at one person, the answer is really obvious. If the variant occurs in 1% of people, our probability if we look at one person is that we have a 1% chance of seeing it. Yeah? And the converse is we have a 99% chance of not seeing it. As you'll see, this is actually the more important part. If we then go up to 2 people, in the first person we look at, we have a 1% chance of seeing it. It's possible we may, when we look at the second person, we may also find the second person also has it. That's really unlikely. It's 0.001 times 0.001. It's like, what, 1 in 1,000? No, 1 in 10,000, sorry. The chance that one of the 2 people will carry it is this plus, sorry, this plus this plus this. if we now look at 3 people, the chance that any one of those 3 people will have the variant is all of these possibilities plus all of these possibilities plus that one. And as we keep going up, you start to see the possibility of any one of the individuals we look at will carry that variant starts to get kind of hard to calculate. As I said, this is the much more easier way of thinking about it. Because at each individual we look at, there's a 99% chance they won't carry it. So rather than ask the question, if I study 4 or 5 or 6 people, what's the chance that one person will carry it? Let's flip that on its head and say, actually, what's the probability that I don't see it? Because that's way easier to calculate. And the probability that I don't see it is, in any one person, 0.99, simply multiplied by the number of people I look at. Well, sorry, multiplied, yeah, to the power of the number of people looked at. Does that make sense? And the nice thing about probability is the probability that we will see, that we will look at people, or we will see something when we look at people, always adds up to 1. There's a 100% chance that people will or will not carry this variant. Is therefore the probability of all these other possibilities where we find at least one person that carries it is simply 1 minus 0.99 times the number of people we looked at. So now that's actually pretty easy to calculate. You could do it in 30 seconds in Excel. 0.99 times the number of people we studied. And so you can easily come up with this table that says, all right, if I look at 2, 3, 4, 5, up to n people, my formula that tells me what's the probability I'll see at least one person with that variant is simply 1 minus 0.99 to the power of the number of people studied. So you can start to see that once you look at 5 people, there's a 4.9% chance you'll see at least one person with that variant. So can anyone guess, if we looked at 100 people, what's the probability we'll see that variant at least one time? I'm guessing a lot of you have the slides open, so you might already know. So it's actually about 63%, which is maybe surprising to some people. You might think, oh, a variant of 1% frequency, if I look at 100 people, like, intuitive, you might say, oh, on average, I have a pretty good chance of seeing it. It's actually, you have a pretty high chance of not seeing it. But then our starting question was, how many people do we need to look at to have a 95% chance? So any guesses? People that are not looking at the slides? 200 to 400? That's a pretty big range. 1 to 1 million. 95. You say 200? So it's actually 299. So when you go up to 1 minus 0.99 to the power 299, that's the number you get. I had a 95% chance that at least one. So now that's 300 people. You're almost guaranteed to see a variant of 1% frequency. So you can then do the same calculation, actually very quickly, for variants of different allele frequencies and come out with a plot like this that you can throw into a grant and say, all right, if I'm doing a study and I want to be pretty darn sure I'm going to find the thing I'm looking for, as long as you have some reasonable estimate of how common that thing you're looking for is, and again, this is where the hand waving comes in sometimes, you can say, all right, with my sample size, I have an X% chance of seeing that thing at least once. So again, that's really good to know for grants, because it impresses people and tells them that you've thought about your study and, is it going to work or not? And it's really good to know conversely, because you can read other people's studies and say, well, honestly, they probably wouldn't have seen that even if it was there because they just didn't look at enough people. Or conversely, yeah, they really did look really hard and didn't see it, therefore a reasonable conclusion is it doesn't exist at that allele frequency.
So that graph is based on by y equals allele frequency to the x.
Sorry?
The graph is made by y equals allele frequency to the x, where each line is the allele frequency.
Yeah, so the different colored lines are the allele frequency in the population. So here is the 1% example we just went through. So once we look at 200 people, we had about I think it's like 86% chance. We go up to 300, you see it hits about 95%. And then the other lines are variants that are more rarer or more common. So see, if you have a variant of 0.1% frequency, you're going to need to be studying like thousands of people to be able to have a good chance of spotting that. So again, these things are just good to like wrap your head around, because you start doing any kind of like population studies or things like that, you know, if you're not aware of these things, you can easily get tripped up or get really terrible reviews for your grant and you don't really understand why. Yeah.
So how would a study about this like start? Would you just find a one-off example of someone who had a strange mutation once, and you want to know if it's in 1% of the population?
Yeah, so okay, so I'll give you the backstory. This is actually a figure I used in the grant. The study we were doing that did get funded was looking for epigenetic mutations as a cause of congenital disease. And the example we took was Fragile X, which was the most common syndrome that's known to be associated with an epigenetic abnormality. So it's this triplet repeat expansion on the X that gets methylated and switches off this FMR1 gene. So we had a figure where we showed, hey, we can detect this with our assay. And we had this and saying, if we hypothesize that there are other things out there that we don't know about of similar frequency, or that maybe are rarer or more common, we're going to study in our grant, we're going to say we're going to look at 200 people, what's the probability we'll see that? And we can say, hey, yeah, the Fragile X, we're pretty sure we're going to see that. So like, we can be really confident that if there are other things out there of similar frequency, that we should be able to see them. So you can start to build, you know, based on some reasonable logic, but there's still a lot of unknowns, but you can, you know, construct some argument that you can use to convince people that the study you're proposing is thought out and has a good chance of success, for example. Yeah? Sorry, can you speak up? Yeah, I don't want to get lost in the weeds, but it's basically just saying, as an example of a disease that's associated with an epigenetic change in the genome, this is one we know about. When you get a full mutation, you get methylation of this gene. You know, and we just use that as an example of if there are other mutations with similar frequencies, you know, would we see them or not? All right, so yeah, this is moving on a little bit to a slightly different topic, and now just talking about p-values, which are, I think, again, similarly important to understand what they mean. So people throw p-values around all the time, you know, and it's sort of in science what our benchmark is of whether a study is successful if we have significant results. But you really need to know, what does a p-value mean, and I'll show you some examples to understand, you know, is that really significant or not? So I would say the real definition of a p-value is the probability that the observed data distribution we see could occur by chance. And nominal significance is usually considered to be about 0.05. So if we were doing a single test, the converse of that is there's a 0.95, 95% probability that the data I saw is real. The converse of it is, however, there's a 5% chance that I may have seen that data simply by chance. And a 5% probability implies that there's about a 1 in 20, like I say, chance that it could have been a spurious association I'm reporting. There's really no link between, you know, cause and this genotype and that disease. It might be spurious. And as I'll show you in a minute, this is a real problem when we start talking about genomics, because usually we're not doing this simple example of one test. We're usually looking at things where we may have tested 1,000 different loci or 1,000,000 different loci. And how do you start to think about p-values in that context where some of the laws that I would say you can usually apply statistics can start to break down? All right, so here's some examples just to sort of start explaining that. So let's imagine we have a coin. You flip it and you can get a head or a tail. And the chance of, on any individual coin toss that you get a head is 50%, 0.5. So let's say if we have that coin, we flip it 5 times, what's the probability that we're going to get 5 heads in a row? It's pretty easy to calculate, just like we did before. It's 0.5 times 0.5 times 0.5 times 0.5 times 0.5, or 0.5 to the power 5. And when you multiply that out, that's about 0.03, or a 3% chance that you're going to get 5 heads in a row. So if we did get 5 heads in a row, standard statistics would say that is less than 0.05, and you would reasonably conclude that that coin, if you just grabbed it, flipped it 5 times and got 5 heads, is a biased coin. Okay? People agree? Now imagine we didn't just take that coin and flip it 5 times, but we took that coin, flipped it 5 times, and then repeated that 49 more times. So we had 50 independent tests where each time we did 5 flips of the coin, and the question now remains the same. Do we think that coin is biased? Okay? Now suddenly the problem's got, well, it's shifted, let's say. It's got more complicated. Because the probability, as I just showed you, of getting 5 heads in a row on a single series of 5 flips is about 3%. But now we're going to repeat that test 50 times. And if you calculate out the probability that in my 50 different series of flips that I'll get at least one of those 50 series will have 5 heads in a row, it's actually pretty likely to happen, about 78% chance that at least one time I'm going to get 5 heads in a row. So up here we said, all right, if we get 5 heads in a row, that's significant, it's a biased coin. Now, what do we conclude? Because it's actually probably going to happen more times than not, but we're going to get 5 heads in a row at least once. So we obviously can't simply say if we get 5 heads in a row at least one time, that's a biased coin. So we need to do something to correct for this, okay? So a way that people often use is something called the Bonferroni correction. I'm guessing lots of people have heard of that. Anyone not heard of it? It's okay to say no. Nobody ever likes to say no, I don't know the answer to that question, but anyway. So Bonferroni correction, the idea is pretty simple. It simply says if I perform many tests, that in order to correct for the fact that we did 50 different replicates of that coin toss, I will divide my p-value by the number of tests performed. And so in this example of where we had, 50 times we did 5 coin tosses, the Bonferroni correction would say rather than 0.05 being my level of significance, because I did it 50 times, my p-value that I need to achieve is now 0.001. In other words, 0.05 divided by 50. There's a real problem there, though, because Bonferroni corrections can be massively overly stringent. Just to explain that, we now said, okay, my p-value needs to be 0.001. In the case of this coin toss, that's impossible now to achieve, because with 5 coin tosses, even if you get 5 heads, I just showed you that the p-value is 0.03. In other words, greater than 0.01 on any one series of coin tosses, I've now made it impossible for my test to be significant. They'll ever, on any one of those series of 50 coin tosses, I can never achieve significance. Okay? And I'm not necessarily going to give you answers here, I'm just going to pose contradictions and make you think. So, just to throw in another fact, I've said previously that when we do genome-wide association studies, people typically take a threshold of significance as 5 times 10 to the minus 8. Does anybody know where that comes from? It's simply a Bonferroni correction based on the fact that somebody once estimated there's probably around 1 million independent linkage disequilibrium blocks in the genome. So we take 0.05 as nominal, divide it by 1 million, you get 10 to the minus 8. Sorry, 5 times 10 to the minus 8. So just an aside, GWAS significance level is based on a Bonferroni correction of an estimate of how many independently segregating bits of the genome there are. Okay, so I've told you that Bonferroni can often be overly stringent, and that's definitely true. It's useful, but you need to apply it with caution. What people often prefer, simply because they're more likely to get significant results, I would say, when I say prefer. Just like everyone likes using standard error bars and not standard deviation bars. It's something called the false discovery rate, so FDR for short. This, like Bonferroni correction, basically attempts to account for the fact that you've done multiple tests, and I would say a lot of people often read too much into it. So for example, FDR, there's a formula that you use to convert your p-values that come out of your test to what's called q-values, and if your q-values are less than at 0.1, according to the FDR calculation, that would suggest that about 10% of your observations at Q values of 0.1 are false associations. So a lot of people, like I say, overly interpret that and say, oh, we only have 1% false discoveries or something if they use Q value of 0.01. I'd say that's bogus. Because really all of these sort of multiple testing corrections, whether it's Bonferroni or FDR, are really just statistical games. Your underlying data is not changing at all. It's the same data. And you're just attaching a different p-value or q-value to it or whatever and saying this is significant or this is not significant. And so it's really just sort of moving the goalposts. I would say often a good way of doing things is looking at your data, applying some of these different approaches and trying to get a good feeling for it. Some studies you'll read will maybe use a Bonferroni correction, another one will use a 5% FDR as their cutoff of what's significant, another one 10% FDR, 1% FDR. There is no magic answer. It's usually people choosing something where they get some reasonable number of data points that hopefully exceed that, and then they have their conclusions. And so really, how stringent you are is again, it's nice sometimes to have known controls that maybe will pass some threshold or some feeling of the data to try and get to some thing that feels right, I would say. And multiple testing corrections can also create paradoxes. So I just gave you one example of coin tosses. Here's another one. And this one often makes people go, huh? All right, so let's say you're doing an experiment, and let's say, again, you're looking at some mice, and you do some RT-PCR or whatever of gene expression, and you're testing this gene in, case versus control. We use box plots. We didn't use horrible bar plots. And we got p-value 0.03. And it says, yes, my affected mice really have higher expression of this gene than my control mice. And you're like, fantastic, let's publish a paper. Okay? But then your boss says, hang on, there's 2 more genes in this pathway. We should test these as well. And so you test them, and there they are, genes B and C, and they're not significant. All right? And now you send your paper off for review, and the comment comes back, and it says, well, you're claiming this significant difference, but you've tested 3 genes. You really need to apply a false discovery correction or a Bonferroni, they may say. And if you apply a Bonferroni, your p-value, because you tested 3 genes, is now, for this one gene, 0.09, because you just tested 3 genes.
Example, displayed your data unfaithfully, because in the real truth of the matter is that you started with one hypothesis, executed one experiment. Then the second one, you actually had another hypothesis with 2 endpoints, executed a completely separate experiment. It seems like
that would be one viewpoint.
That would be. I think I'm just curious from like a statistical standpoint. It seems like statistics have to be very rigid about figuring out which statistical test to apply before you actually, during your design phase, before you get your results, which is never how it happens in real life.
I was going to say that's what you may be told in theory, but in reality it doesn't work that way. And yeah, the other thing I would say, you talk to 2 or 3 different people, you probably get 2 or 3 different answers.
Or 4.
Yeah. So yeah, I'm just putting this example up to more be provocative than be statistically correct. You may say, oh, this is hypothesis 1, and that's a different independent, I don't care. Let's imagine you tested 1,000 genes then, all right? Same point still applies. But basically, you can create paradoxes where sometimes if being too greedy or testing too many things, something that if you had a narrow focus would have been deemed significant is now no longer significant. And I put all that in quote marks because is that really true? Is that really what it means? And again, I don't necessarily have an answer for you, but it's just these are good things to think about and be aware of what does a p-value really mean? And a lot of people often present their data of these are real and these are not real when things are just slightly one side of a threshold they impose on the data, and they're now converting some range of values into a binary yes, no, truth or not truth. Anyone got any thoughts? Yeah?
Do you ever run across in these kind of studies the other example where you have a study that's so large you're going to get a p-value that's significant no matter what?
Yes. Nice introduction to my next slide. So yes, so this is exactly one of the problems that you get in genomics sometimes. That a lot of statistical tests that are commonly used were really designed for, moderate sample sizes, let's say. That's where they work best. If you start throwing in enormous data sets into them, they will give you massively significant p-values that really has no useful bearing on your data. And this is exactly an example of that. It's real data. I wouldn't suggest anyone do this because I'm kind of throwing it in there as again, another provocative example just to illustrate the point, but let's look at it. So here I took chromosome 1 and chromosome 2, the two largest human chromosomes. I said, what's the length of their euchromatic sequence? Euchromatic being key there because chromosome 1's bigger than chromosome 2. It just has a really big centromere, so less base pairs in the euchromatic sequence part. And then said, what's the number of SNPs on these two chromosomes? What's their fraction? And so on chromosome 1, it's 4.5%, or 4.58%. On chromosome 2, it's 4.6% of the bases are labeled as common variants on those two chromosomes. Here it is appropriate to use a bar plot because it's a single value, it's not a range of values. But there we go, we plot it, and it's basically the same, okay? But if we now apply a statistical test to that, and a chi-squared would be an okay test to say, is there a significant difference in the density of SNPs on those two chromosomes? We get a chi-squared statistic of 6.3 times 10 to the 16, which is impossible to calculate what that p-value is. It's bigger than the floating point of r, p-value less than 10 to the minus 368. Is that useful? I mean, that's really great in a paper, a p-value of 10 to the minus 368, but it doesn't mean anything. Like, it's just because we threw in so much data, it swamped the statistical test, and there is no difference. Okay, so p-values can kind of work both ways.
I don't know whether I understand correctly or not. Just sometimes I feel like it's not the statistical test itself have some problem. It's just the way we construct the null distribution have a problem. Because when you're doing the test, you have to clear what is our null hypothesis and null distribution. For example, the last example, a gene expression. When we're doing that one gene test, the null distribution is you do thousands of experiments for this gene under two conditions. Then you construct a distribution. Then the p-value less than 0.5. That is your statistically significant.
So you're saying permutation testing?
Yeah, but when you're doing within one.
This is great. You're like introducing my slides to me. Yeah.
If you're doing testing for multiple genes within one experiment, that is not how would you using the statistical test or t-test? It cannot use it that way. It can only apply to your one simple experiment or one test on one simple question.
Maybe, but yeah, now we're in a world where most people test a lot of genes at a time. Like you run an RNA-seq experiment, you're going to get data for at least 10,000 genes in any one tissue. So we have to come up with ways of dealing with that. Most commonly, people use FDR because it's some reasonable, it's not overly stringent like Bonferroni often is. Sometimes you can apply Bonferroni and still get significance after Bonferroni correction, and most people go, okay, you tried really hard and it's still significant, I believe you. Most people usually go with FDR because you're not usually in that situation. It's a reasonable compromise.
Give you an idea of your machine, how your machine is accurate. Because you using one machine, doing one experiment, you get expression for different genes. Then if the p-value like FDR less than 0.01, 0.05, that means how your machine performance. I think that is the way to interpret the results. It's not about the gene, it's about your technique. Or
Maybe, maybe not.
Yeah, what's the chance of your technique give you significant results? It's not how your gene biologically differentiate.
Well, maybe. I mean, that's more coming into then experimental design. How many samples do you need to look at to get robust data? And again, I'd say there's lots of papers that look at like 2 mice with a disease and 2 mice as controls. And for me, that's like, okay, they're inbred mice, but come on, still. You repeat the same mouse 5 times and you get 5 slightly different answers. Like, any other thoughts? Yeah?
How do you know when sample sizes get too big? Like, what's the threshold?
Don't usually see that, to be honest. I mean, this is a ridiculous example that I chose just because there's millions of base pairs on a chromosome, but that's maybe getting into the realms where it is okay, but yeah. I mean, a lot of these tests work best in the order of a few hundred or a few thousand, which is typically what you have in science.
That's the wrong question. The question is, what are the effect sizes too small? Because
yeah.
What people forget about significant values is that you can get something that's significant, that's half of it. The other half is what's your effect size. If it's not going to be relevant to your system, in my case it's always is, if it's not going to be clinically relevant, then I don't care.
Yeah.
And so
it's kinda
being aware of both the effect size and the statistical significance, that's when you begin to answer, not when you have too many samples, but when you start looking at significant differences and you go, that may be significant statistically, but it has no bearing on the behavior. It probably has no bearing on the behavior of the system compared to another factor because the effect size is so much.
Yeah, it's kind of like this plot. So exactly, effect size is the x-axis. So imagine we had 4 million individuals. We basically have a line that went like that. And it's going to say, even if we have minuscule effects that are influencing 0.1% of the variance in some data, which is smaller than your technical variation of your experiment or something, it's still going to come up with a huge p-value if you have an enormous sample size. And that's exactly what we have here in this stupid example I show you of the chromosome. It's giving us a massive p-value, but it's merely a fact that we put in so much data, we have such a huge power because we have so many observations that even a minuscule difference is coming up with an enormous p-value. And so is the p-value meaningful anymore? Maybe a fold change would be a better statistic to represent this. And then the fold change is going to be minuscule. It may still be highly significant, but that's, in this context, probably the more meaningful summary statistic of this data, not just labeling it, sticking a p-value on it. So yeah, usually I like to always show fold change and p-value with something. And if you had 10 samples and 100 samples and 1,000 samples of the exact same fold change, would have very different p-values. So I think it's always most meaningful to think about things in both of those two terms, the fold change, the effect size, and the significance of that. Because the significance is really strongly driven by how many samples, how many observations you have. And that's why GWASs need massive sample sizes, because the things that they're going to see have small effect sizes, because common variants don't have very big effects on disease, because otherwise they wouldn't be common variants anymore. You wouldn't have variants that are really common that are affecting whether we die at age 20 or something. They don't exist. They get weeded out of the population. But yeah, all these things, like when you start to understand them, I think you can start to interpret science in a better way, a more rational way. So yeah, I think this is kind of the point you were getting at, that there are other ways besides maybe chi-squared or things that are more appropriate to start to apply to some of these cases, particularly that we come across in genomics, where we may have large numbers of observations or distributions of data that don't fit well with the assumptions of many statistical tests. And so permutation testing is something that I think is probably one of the best ways of testing significance of many genomic data sets. The basic idea here is that rather than putting your numbers into a standard statistical test, in permutation testing you're simply taking the data you have, scrambling it, and asking, if I scramble it, how often do I see the thing that I really saw? And so there's very little assumptions based in that. You're not assuming your data is normal or non-normal or anything. You're just saying, okay, if I randomize it, did I see what I saw really? If I randomize again, did I see it now? If I randomize again, did I see it now? You repeat that 1,000 times, 10,000 times. And then you can build up a null distribution of what would I have got if my data was really just random, and then ask the real observation I saw, did it fall in the way in the tail? I.e., is that unlikely to have occurred by chance? So let's take the example of, SNP density on chromosome 1 versus chromosome 2. The really important thing here is the randomization, because while permutation testing is a really good way of getting a handle on significance, what was the likelihood that the observed data could have occurred by chance, you have to be really careful that when you're randomizing your data, you're not introducing some new bias that influences your permutation test. So in the case of this SNP density, rather than taking all these SNPs and then just randomly saying, well, we had 2 chromosomes, so we randomly assigned chromosome 1 or chromosome 2, that's going to create a bias if I did that. Because even though I had 2 chromosomes, the 2 chromosomes aren't of equal length. Chromosome 2 actually has some more euchromatic sequence on it. It's a bigger fraction of the total search space than chromosome 1 is. So in this case, when I take my 2,147,000 SNPs that I have, I would assign them to either chromosome 1 or to chromosome 2 based on the fraction of sequence that each chromosome represents in the total of 1 plus 2 added together. Does that make sense? Because if I just assign them 50-50, I'm going to end up with exactly the same bias I started with, or I'll create a new bias. And so we would do that. We'd take all our SNPs, we'd assign them to the 2 chromosomes randomly, but incorporating the amount of space they each represent. We build up a null distribution of the number of SNPs, say, present on chromosome 1, and then ask what was the true observation we saw. And if our true observation lies in one of the 2 tails, again, you have to decide where your cut is. Is it in the 5% tail, the 1% tail, whatever? You would then, based on that, assign whether is there really a statistical difference in the SNP density on these 2 chromosomes. And so your p-value is determined by the number of random permutations that equal or exceed your real observation. So say we did 1,000 permutations, we shuffle the SNPs 1,000 times randomly assigned to the chromosomes. If 17 of the 1,000 times we did it, we actually saw equal or more than this fraction, our p-value will be 0.017. So I'm kind of skirting through this because just to give you a sense. And that was actually my last slide. So I just hopefully have given you guys a bit of a flavor of some of what I think are some of the important things when you're starting to think about genomics. And yeah, if there's any other thoughts, then pipe up. It's nice that people chipped in with questions. Yeah?
So maybe a lot of people already know this, but the American Statistical Association released a statement on p-values this year, kind of urging the scientific community away from the 0.05 obsession and kind of urging people to know the appropriate definition of p-value. And I would just recommend reading that.
Yeah. Any other thoughts? No? All right. Well, thanks, guys. That's it.